throughput. Okay. So we're going to talk why microfluidics are important, why throughput is important, and how you can build your own microfluidic device for your own experiments if you want to do research. We're going to talk a little bit about bioapplications in microfluidics and also at the end, microfluidics limitations. Okay, so microfluidics is the science and technology of systems that process and manipulate really small amounts of fluids using microchannels. So if you can imagine, if you have a ruler, you get 1 millimeter of this ruler, there's 1,000 points in just 1 millimeter, and you build channels out of this. And basically the channel have an input, an inlet, so you can pass flow through, then a channel, and then an output. So basically, if anyone just want everyone to participate here, this is what you can see at the bottom is a microfluidic device. So if anyone can annotate and show me where do you think is the channel and the inlet and the outlet in this device down there. So I'm going to give you guys a minute if you can annotate. Okay, cool. So most of you got the channel right. The channel is this part here. And the inlet and the outlet, this is the inlet and this is the outlet. And now the reason why the inlet shape is different, we're going to discuss this in the fabrication slide. So just, you know, we'll go back to this later. Cool. Now let me go to the next slide. Awesome. Cool. So I want to talk about since we're already also talking about cell models, 2D cell models, 3D and mouse models, we want to talk about the scalability of these cell models and how microfluidics can actually help in increasing the throughput of these cell models. So let's start with the first cell model, which we had a question about, the 2D cell model, which you are going to culture it manually, and then you're going to sort it up until recently manually, right? There's an organoid model, which you can basically also culture it manually and sort it manually. Of course, with mice, it's really hard to automate how you sort mice unless you guys saw the Boston Robotics. Who can you raise your hand if you saw the Boston Robotics? Maybe they can basically build some sort of a robot that can, you know, sort. Awesome. Okay, Nathan, good. So yeah, you can see basically you can build a robot that can sort these mice accordingly. But let's just now focus on 2D, right? So up until recently, just 4 years ago, we were actually culturing these cells manually and also sorting them manually with a micro manipulator. But then microfluidics came along, and now let me see if I can go to the next slide. Okay, so now there's with microfluidics, we were able to automate these single cell sorting devices, and that really increased the throughput of 2D cell cultures. So if anyone have any idea of what is the microfluidic technology that really scale the throughput of single cell analysis, you can raise your hands or you can type it or say it. Okay, Gabriel.
Was it FACS?
Yes, awesome, great. So it is FACS, developed by BD in the 70s. And it is actually basically a microfluidic device. You flow the cells in a channel and then you image it, and then based on a fluorescent signal, you sort it. So, and this is basically a microfluidic technology. Now, who can tell me what is the current, why we have now a current urgent need for automated high throughput tests? We are kind of living it in the moment right now.
Coronavirus.
Yeah, right? So with the coronavirus. And if you think about it, one of these really high or rapid tests it's actually a paper-based microfluidic. So even this paper, how you design the paper mesh inside to put your blood sample or any other samples inside, and you can basically get some antigens and antibody to reaction in this device. It's also based on biomicrofluidics. So there's really an urgent need for increasing the throughput and also automation. Now, if I'm gonna have a breakout session, I think Ali know how to do this. Also, I want you to discuss what do you think in your mind is the really important biological application essays that we need to increase the throughput in this or the automation in this. And I'm gonna give you guys 5 minutes. For now, throughput, let's go back to the lecture. Throughput is really important, as some of you might mentioned already, is that it's better to get a statistical significant data. And sometimes throughput is necessary. It's necessary to increase your sample size to get the right answer. So there's actually an equation for this that you can use in any of your experiments to see if you will get, what is the sample number that you need to get a statistically significant data. And so basically you get the N is the population size, the critical value of normal distribution. And as you may know, some of you, if you want to submit your data to a journal, they won't accept the critical value less than 95% confidence level. So this is really important. And then you get the sample proportion, and of course the margin error. Of course, margin of error is usually like a human error, right? So based on these, your test, based on your assay, even with COVID-19, even with any type of assay, they usually use this equation to know what is the number of DNA I need, what is the number of cells do I need just to get this test right, or have to get at least a 90%, 95% confidence level. So, and this is basically, you can apply it also in your experiments in the future. So let's now shift gears a little bit and talk about also the current technologies for, I'm gonna focus more about single cell sorting and how we can use microfluidics for this. So you can see in the x-axis is the throughput, right? And we discussed how much important it is just a bit. So what is the number of cells you can sort per second, right? And now in the y-axis, you can see what is the information that you want to get per cell. As you mentioned in the facts, they only care about positive or negative, right? Whether the cell have the protein or not, whether the cell have the receptor with the fluorescent tag or not. But of course, now we will discover more that cells are heterogeneous, even within the same cell population, even with the same cancer cell line, there are subpopulations. So now we want to know also, not even if that protein is there or not, we want to know where it is in the XY coordinate inside the cell, right? And not only that, we want to know more about what is the distribution of that protein in the cell. And of course, at a really high level, we want to know, for example, what is the concentration of that protein in a certain amount of time, or as a function of time. What is how the expression of that receptor in a certain amount of time, because it's really dynamics, a lot of dynamics that happening within the cell in protein expression, and of course, all the other processes. So let's see the picture on the left here, the well plate based pipetting. This is the most ancient way of processing or sorting single cells, right? You get single cells in a well plate, and then basically you pipette them out just to process them or sort them. Now, why does this method have really limited throughput? Now, for the interest of time, I'm just going to answer it. It's just because it's limited by the well plate, right, numbers. So you have 96 well plates, you would have 96 cells to process in that amount of time. But with microfluidics, you can actually flow a huge number of cells, a million billion cells in the channel and process them in a nanoliter, right? So this is why microfluidics steps in and really increases the throughput of single cell sorting and analysis. And this is the way to go. If you want to increase any sort of assays, you have to go to microfluidics, especially for cells protein, DNA, RNA, which is at this scale. So, okay, so now you have this idea, right, that we discussed in the breakout sessions. You have many ideas. Okay, now how can I make a microfluidic device for this? So the steps in developing microfluidics for bioapplication: first, you have to define your bioapplication requirement. So what do you need? What is your cell size? What is your cell concentration? What kind of reagent do you need in your application, right? And then we're going to go to the design of the microfluidic channel. And this is what we're going to discuss in the next slides. And then after you design your microfluidic channel, because you don't want to really do trial and error, right? You don't have the resources, you don't have the time, so you have to really build a really fit a microfluidic channel for your bioapplication, and then at the end you can redesign it if it doesn't, or you want to improve it at the end. So, and then you were going to talk about microchannel fabrication, and then we're going to move in. After you fabricated your channel, now you're going to test it on your cells. And then you have to, of course, test the cytocompatibility, because not all cells respond, cells respond differently to the channel size or the shear inside the channel. Or if for one part of the channel, the cells won't like it because it have high pressure. So you need to really redesign then at the end redesign your channels based on your bioapplication requirements. Okay, so let's go now step into the first part, which is the microfluidic principles.